This is a video to help you through the activity on phospholipids and bilayers. In front of you is the chemical structure for a phospholipid and the chemical structure for a triglyceride. We've talked about triglycerides in a previous activity. Now we're going to focus on phospholipids. The first thing I'd like you to do is notice the things that are similar and different about phospholipids and triglycerides. Both of these molecules have a glycerol core. We learned in a previous activity that a triglyceride is formed from glycerol attaching to 3 fatty acids through condensation reactions. The same thing is true with triglycerides. There is a glycerol core and there are fatty acids attached. I'll circle the glycerol on both molecules and the fatty acids on both molecules. One difference is that a triglyceride has 3 fatty acid tails, whereas a phospholipid only has 2 fatty acid tails. Instead of the 3rd fatty acid, like a triglyceride has, the phospholipid is attached to something else. One of the most important parts of this new group is that it's ionic. It's called a zwitterion, which means it has both positive and negative charges. It has a positively charged primary protonated amine, and it has a phosphate group. This is an organic phosphate group, and the most important quality of this group is that it's ionic. And that is going to drastically change the interactions of a phospholipid with water compared to a triglyceride with water. As you already know, a triglyceride is the molecule that is in fats and oils. These molecules are overwhelmingly nonpolar. Despite the fact that they have some polar groups, the nonpolar groups overwhelm the characteristics of this molecule. Triglycerides are not water-soluble. Associate the word triglyceride with oil or fat and remember that these things are not soluble in water. Despite some polarity in this molecule, it is overwhelmingly nonpolar and it is not water-soluble. You should know this from experience. And if you don't, go to the dining hall and try to dump some oil into your water glass and see if they mix together. A phospholipid has a polar ionic group on it. The phospholipid has a nonpolar part, fatty acid, hydrocarbon tails, but now its polar part is ionic. So it has the same polar groups that a triglyceride has, but now it is this ionic group in addition to that. This ionic group changes the balance. Now the ionic part is polar and soluble in water, whereas the fatty acid tails are nonpolar and not soluble. This is an amphipathic molecule. It has characteristics of both polar and nonpolar parts that are important for its behavior in water. On the next slide, I'm going to compare a phospholipid to one of its fatty acid components. We've already discussed fatty acids and their interactions with water to form micelles. Both of these molecules are amphipathic. They have ionic parts that interact strongly with water through ion-dipole interactions. And they also have the nonpolar hydrocarbon parts that will not interact favorably with water and will be excluded. So both of these molecules have very similar properties in the fact that they're amphipathic. They behave a little bit differently in water, and the difference mainly stems from the overall shape of these molecules. We saw that a fatty acid is kind of triangular in shape. It has a large polar head group with an ionic charge, and its hydrocarbon tail is a little bit thinner, so it behaves as a triangle in solution. We could draw a cartoon that looks like this, and you saw me draw something like this on a previous activity. When a lot of these things assemble in water so that the polar head group interacts with the water and the nonpolar tail is excluded, you end up forming a sphere because these triangular or cone-shaped groups end up having a larger outside and a smaller inside, forming a sphere in water, where the outside is polar and the inside is nonpolar. This is a micelle. The phospholipid has a different overall shape and it forms something that's similar to a micelle, but it's very different in shape. The phospholipid has a polar head group and hydrocarbon tails, but its overall shape is more rectangular or cylindrical. On my paper here, I've shown the two-dimensional shape. In 3 dimensions, this one's more like a cone, and this one's more like a cylinder. But we can use the two-dimensional shape to represent what happens. If you line up a bunch of these phospholipids next to each other so that the polar head group can interact with water and the nonpolar tails are away from the water, you end up getting a bilayer. It looks something like this. Here's one phospholipid group, a cartoon for the phospholipid. The polar group is represented by the circle, and it's going to have ion-dipole interactions with water. Very strong attraction. The nonpolar tails, which are represented by these 2 squiggly lines representing the 2 fatty acid tails, are away from the water. This makes a layer. Basically, all of the polar head groups are facing the water, and the nonpolar tails are facing away from the water. Another layer forms on the opposite side, so that now you have something called a bilayer. These are the things that make up cell membranes. Here are pictures of 2 different types of cells. All cells are similar in that they have cellular membranes. They have to separate the outside solutions from the inside of the cell. Both of the inside and outside solutions are water solutions, and they're hydrophilic or polar solutions. The cell separates the inside and the outside using a phospholipid bilayer. This is the cartoon from your book that represents a phospholipid bilayer. The polar head group is represented by a circle, and the fatty acid tails are represented by 2 squiggly lines. In the center of a phospholipid bilayer, you have a nonpolar region. This can separate an aqueous polar environment from the outside of the cell and the inside of the cell. Here's another representation. This could be the outside of the cell where there is a water solution. The hydrophilic head groups of the phospholipids are represented in this bilayer by gray circles. The bilayer's fatty acid tails are represented by these orangish color squiggly lines. This is how a phospholipid bilayer can separate the water from the outside of the cell to the water solution of the inside of the cell. And here's another cartoon. Once again, the phospholipid head group is represented by the circle, and its fatty acid tails are represented by squiggly lines. In this cartoon, the cell membrane is represented as the green membrane on the outside, and this cell is taken as a cross-section where you can see the aqueous or water environment inside the cell as the blue material, and the nucleus is this gray ball. The membrane lines the outside of the cell, and it separates the inside of the cell from the outside of the cell using this phospholipid bilayer. In this cartoon, there are other things that are shown in the cell membrane. One is an integral membrane protein. These proteins are used to pass things from the outside to the inside or from the inside to the outside of the cell. It's essentially a tunnel through which polar ions or small molecules can travel through this cell membrane. Another thing that I'll note is the yellow blob here. This is representing a cholesterol molecule. Cholesterol is important for the fluidity of a cell membrane. Let's look at the structure of a cholesterol molecule. The cholesterol molecule has a basic steroid ring structure. It is a steroid, just like testosterone or estrogen. All steroids have this basic ring structure, 3 6-membered rings and a 5-membered ring. Notice that you see the same pattern here in cholesterol. This is the chemical structure of cholesterol. It has a very large hydrocarbon part and 1 alcohol. Cholesterol has a nonpolar part and a polar part, so it is characterized as a lipid. Cholesterol embeds itself in the cell membrane by interacting its polar head group with water and its nonpolar part with fatty acid nonpolar tails. I'm going to draw a cartoon of cholesterol like this. Here's my cell membrane or my phospholipid bilayer with the polar head groups lining the nonpolar interior of the bilayer. Water would be on the outside and on the inside of this membrane. And the cholesterol molecules are attracted to water and the polar head groups through the alcohol group, and they are pointing their nonpolar parts away from the water and lining them up with the nonpolar parts of the phospholipid. Cholesterol is important for the fluidity of a cell membrane. If there's not enough cholesterol in your cell membranes, the membranes are normally too flexible. And if there is too much cholesterol in cell membranes, the membranes are too rigid. So there's a happy medium here where you have to have just the right amount of cholesterol in your cell membranes. And there are lots of biological processes that regulate this. You obtain cholesterol through your diet, and your liver synthesizes cholesterol. So you don't have to eat cholesterol in order to have it in your cell membranes. The cholesterol in our diets are not necessarily related to the amount of cholesterol in our blood or in our cell membranes. There's a link below this video if you want to learn more about this. Here's a picture again showing the cholesterol incorporated into the cell membrane in this cartoon. Also notice that there are these proteins that are aligning themselves in the cell membrane. The proteins that span the entire cell membrane need to have polar groups on the two ends of the protein so that those polar groups will interact with water. And then they must have nonpolar groups on the outside of the protein where the nonpolar groups would interact with the fatty acid tails of the phospholipid bilayer. If I would want to draw a cartoon of a protein like this, I might draw this structure, where here's my protein with a nonpolar center on the exterior of the protein and polar groups on the exterior of the ends. These proteins align themselves with phospholipid bilayers like this, where the polar ends match the polar ends of the phospholipids, and the nonpolar center matches the nonpolar hydrocarbon tails. Notice how these structures are organized and made based on attractive forces alone. Attractive forces between polar groups and nonpolar groups with water are enough to make these structures form. This is the chemistry of the cell membrane. Thanks for watching this video on phospholipids and bilayers. If you have any questions, please post them in the comments. And if you found this video helpful, please let me know by giving me a vote.